Many like me have encountered dark kernels on corn cobs or brown spots on bananas. What if those spots were artificially removed? Would that make the food safer? Because the corn we eat today may not be corn but modified brand P0115, this video will explore food safety of genetically modified foods in order to answer our questions. But what is genetically modified or GM food? We've all heard about frankenfood, food altered with genetic information influencing specific characteristics like larger strawberries. But the average consumer may know less about how this change occurs or what this information is made up of. It comes down to the genes, the traits we carry permanently, passed down from our parents. Genes are strands of DNA which code for particular types of protein. Proteins are much more than the nutrients we get from eating meat or peanut butter. They are made up of base pairs like these, and they determine physical traits like blue or brown eye color. These letters are the genetic code for all of our hereditary information. They define us. Genetic modification replaces base pairs, altering food for favorable characteristics like size or color. Many GM foods are created by a process called transgenesis. Using microscopic needles, scientists transfer genes from other organisms, like soil bacteria, into the crop. The result is a new organism, like modified brand P0115. Now that we understand how genetic modification works, we can turn to the ways in which their safety for consumption is assessed, primarily by the Food and Drug Administration. In 2001, 4 years after GM foods were sold, the FDA regularly tested GM foods for their similarity to food toxins like allergens, for example. They would compare the proteins, and therefore the base pairs, of known allergens to those of GM foods. Such a comparison refers to a process called sequence homology, the genetic similarity between two organisms. We will focus on sequence homology and food allergies to investigate food safety assessment. Now, in order for this assessment to be effective, industries must verify the exact changes of a modification. In 2005, a United Nations organization, the HESI, highlighted the following FDA requirement for GM foods: a description of the purpose or intended technical effect of the new proteins. Here we can read further the intended effect of the changes to new base pairs. But how much control do we have over new proteins and base pairs? Are unintended, undocumented, and unassessed changes occurring? This sequence homology study demonstrates the control we have over assessing foods. In step 1, 6 species of a flowering plant named Thale cress were genetically modified and grown for near 30 days alongside unmodified Thale cress. In step 2, proteins were separated and made more observable by gel electrophoresis. In step 3, the total number of proteins were counted and averaged for each species. In step 4, the totals of modified species were compared to the unmodified version's total, 429 proteins. Although the samples showed differences, scientists Martin Rubelt and Marcus Lipp, along with others, said that there was little variation and no significant differences. If these values had compared modified Thale cress to a food allergen, we would say they are similar and argue that Thale cress is unsafe. Now in step 5 of this study, the same scientists acknowledged that the assessment was too complex. They stated that it requires more studies before being used for food regulation. The process becomes so complicated that scientists admit it doesn't provide an adequate, controlled assessment. In fact, there is no foolproof assessment, and scientists continue to debate about the limitations of GM food assessment. Find out more about safety assessments by contacting the Food and Drug Administration, the Environmental Protection Agency, or the United States Department of Agriculture.